Okay. I can't see how many people logged in. 48. We are 3 minutes over the starting date. This is a session for advances in copying and COP systems modeling. My name is Untitled Chapter. I will be the moderator today. I also will present one of the talks. I am with the University of Florida. I'm also one of the executive committee members of AgMIP. And at Florida, I'm also the director of the Florida Climate Institute. So I assume most of you heard the introduction from Alex and Rohan, that within this session, we actually will talk about the underlying models and their progress, which sort of help AgMIP and the different parts sort of looking at the assessment and adaptation and mitigation options. So today we talk about advances in COP systems modeling. We have 6 presentations, including Monse Salmeron, Mark Kobiels, Dave Gustafson, Lev Siska, and Cheryl Porter. I will introduce each of them when we get there. What I'd like to ask each of the speakers is to stick to the 10 minutes that we actually have, maybe 5 minutes for discussion right afterwards, and also maybe have a few minutes in the very end, sort of more general discussions. So please stick to the 10 minutes. One way to indicate that it's about time, I probably come back on my screen. You probably will see me. So with that, one more thing, I'd like to ask everyone maybe to switch off their screen for the moment, if whoever's not speaking. If you have questions, please add them to the Q&A or in the comments, and we try to.
Senthold, you're muted.
Yeah, I got unmuted. I got muted somehow. I will start with the first presentation, talking about modeling the big 4. And what I try to do in here, I want to talk about 3 of the big 4s, which is wheat, which is the one which I co-lead with some other people, but I also briefly want to mention progress in the teams working with maize and with rice. And I leave out soybean in detail here because we have a presentation today also on soybean. And I'd like to spend maybe a couple of slides on actually comparing crops, see what we learned when we actually look across these different crops. So in AgMIP wheat, it's one of the teams which started almost at the same time when AgMIP started, or shortly afterwards, it's 10 years ago. And I got quite a few things achieved. I just want to briefly summarize these. We started off looking at model uncertainty. We identified within the AgMIP wheat team that the temperature response causes the largest uncertainties. We also looked at how many models you actually need if you have many models available to sort of get increase your certainty to a certain degree. We looked at CO2 impact, and I highlighted here in red, not just with the model, we also had a lot of data, very detailed data from field experiments. We looked at heat impact, including heat shocks. We heard today in the introduction that one of the emphasis during AgMIP8 is to look at and try to understand and build in shocks. So for wheat, we actually had data for heat shocks. We also looked at canopy temperature, and again, using many field data. We did look at adaptation for heat and derived heat stress traits from field data. And most recently, we expanded, instead of not just looking at biomass and yield, we also started looking at the quality issues and grain protein. Again, we had a lot of field data, a lot of data available here where we could look at the impact of CO2, heat, and water. So I highlight data testing because I think it has been a very important part of AgMIP, and it's not just in wheat, also in the other programs, that the modelers link actually very closely with experimentalists, which are so necessary to really understand do our models work? If they don't work, what else do we need to do with the models to get them to work? Here's one example where we looked at grain yield and grain protein. On the left-hand, grain yield, right-hand, grain protein for different treatments with a normal sowing. These are data from Australia. Normal sowing with high CO2. The late sowing creates more water stress. In the first treatment, there's high CO2 plus irrigation. Then the irrigation was taken out. You see how the yields actually dropped before they come up again if CO2 is added to the water stress treatment. You can see the observed data in sort of a yellowish with an error bar, and green are the simulated data. These with an error bar representing the spread of the models. And in general, we get the response right for yield. With higher CO2, we get an increase in yield. We lose quite a bit of yield under water stress, and we make some of that up again with higher CO2. When we look at grain protein, we see exactly the opposite, where with high CO2, we get lower grain protein concentrations. Under stress, water stress, it might come up again. So the model, and you see, we are definitely within the range of the observation. So the models are actually able, at least ensemble of the models is able to reproduce what we've seen in experiments. But we also use these models obviously within the groups for application. As I said, these models are actually a basis for application in many other parts in AgMIP. And I wanted to show you one example where we run many models. I think in this case 18 wheat models across 60 locations for each of them 30 years. So each of these points is a median of 18 models for high rainfall locations, high rainfall and high yielding locations, which are the dark brown symbols. The more yellowish ones are for low rainfall location. What you can see is that the protein amount, the grain protein yield, goes actually up with increasing protein. So you actually, in general, there you can see there some of them it actually goes down, but there's a general trend that grain protein amount goes up, whereas the grain protein concentration, that's actually the quality aspect of grain protein, in general goes down with increasing yields from higher CO2. And this is quite important. You see there's quite a lot of noise on there, but there's a clear trend in there. This is quite important. That's why I showed in here. It's consistent with the meta-analysis by Breakout et al. A few years ago, where they looked through all measurements they could find for wheat and found the same, that the protein amount in general seems to go up, whereas the concentration with higher CO2 goes down. For the future within, or for the next step within AgMIP, wheat, the group is looking at improving traits and really have data again where different traits have been measured to increase yield. There's also an activity now getting started to look at ET. Let's briefly talk about AgMIP maize. Again, similar approach earlier on where the group looked at uncertainty, found similar things in terms of temperatures has the largest uncertainty. They looked at CO2. They did a really great job with lots of data again on ET. And they have an activity now, and we hear more during the panel on low input systems. And for a next step, they realized in ET that in general the models could do it, but there's a huge spread and there's more needed there. So they actually found more data to continue this. And I just want to show you one example, which probably is also shown in a parallel session by Bruce Kimball at the moment. Black are the observed ET rates per day after the days of planting. And then 4 different simulations. Again, these are ensembles of models with different detail. And it's more detail you give the models. In many situations, they get closer to the black line, to the observations, but there's still problems there. And that's one of the reasons this group wants to dive deeper into what's going on with ET, how can we better deal with this. Briefly, AgMIP rice. Again, we shared all the protocols early on, and it allowed us actually to compare the different modeling results. They also found that the T response seems to cause the largest uncertainty. They looked at CO2. I think I have a figure actually here for 2 different phase experiments, one from Japan, one from China, and they also had some SPA data. And you can see that there's a large spread in this case and in all of them in the simulations. There's also some spread in terms of the modeling, but as a median, again, the model ensemble seems to be actually getting quite close to this. They're planning at next steps, looking deeper into elevated temperature, cultivars, different cultivars across locations, and also trying to explore hypothetical traits of how they might be helping to adapt to climate change. One of the big outcomes across, when you look through all of the different models that's documented in with many papers for wheat, maize, rice, also for potatoes, which I haven't mentioned here at all, is that the multi-model ensemble median is a better predictor than any single model. And if you want to know, there seems to be also a statistical explanation now. Please have a look at the paper by Wallach. The last slide I want to show is what we also did is we looked across the 4 main crops. We started first with wheat, but then did it also for rice, maize, and soybean, looking at the temperature impact at different scales. This is at the global scale. And we were able to show a mean of different methods, basically on 4 methods, gridded simulation, point simulation. We also upscaled here observations and 2 statistical methods put together as one method. And you can see that when you take the error bars into account, they actually come all to a very similar result. Maybe a bit more for wheat. There's a bit more spread for rice. Again, for maize. The observations seem to be showing actually more negative impacts on a global scale when you look at the observations. You can also see we have less data for soybean. That's why I think I'm looking forward to see the soybean talk today as well. With that, I'd like to stop here, stop sharing, and open up for maybe a couple of questions if you have. I can see we only have the chat space there. Is there anything in there? If not, I don't want to wait too long here. Keep your question maybe for the final discussion, and let's move to the next presentation. And we might be able to bring the big 4 presentation back into the discussion again. So our next brief presentation will be by Monse Salmeron. Monse is with the University of Kentucky, and she will talk today to us about ensemble modeling applied to new crops, and I think it's be particular about soybean. Monse, over to you, and maybe switch your screen on, and I switch my one off. Switch your video on, please.
Thank you, Senhol. Can you see my screen now and hear me?
Yes. We can, yes.
Okay, great. Well, good afternoon, everyone. And thank you for the invitation to share the work we have done within the AgMIP soybean team. Why did we decide to focus on soybean as a new crop? Well, soybean is the most widely grown legume with 125 million hectares worldwide, and as such is one of our main sources of protein with an expected increase in yield and protein demand as population increases. However, future yield projections show the largest deviations from yield trends by 2050 in a gridded multi-model intercomparison when compared with other major crops. This large variability in model projections might be associated to differences in how models respond to an increase in temperature, increase in CO2, prediction of evapotranspiration and water stress, soil water dynamics. Today, there was no coordinated multi-model evaluation with observed data on evapotranspiration, high CO2, or a multi-model sensitivity analysis to different environmental variables that could help understand how current models differ and what are the sources of uncertainty in yield projections. In addition to the processes I mentioned, nitrogen fixation is a process unique to legumes that will be unique to legume crop models that might influence soybean yield projections. Soybean has a seed of high protein concentration, and as a result, it has high amounts of nitrogen that are exported in the seed each year. In this table, you can see some estimates for nitrogen exported in the seed for US average soybean yields and for my trials in Kentucky last year. So as you can see, soybean has a high nitrogen demand, and research shows that 25 to 75% or more of the nitrogen demand is usually met by biological nitrogen fixation. We also know that this process is highly sensitive to environmental conditions and water limitation, more so than other plant processes, which can influence the ability of nitrogen fixation to meet the nitrogen demand. So soybean is produced mainly for protein, also for oil production, and it is particularly important for this crop that we do not focus only on yield, but also on grain quality. And in this context, I want to share this figure that shows the historical yield trends and protein trends in the US. There's currently concern over the decline in seed protein concentration over time that is, has been associated to a combined effect of genetics and increasing yield potential. However, today there's limited, it's critical that models are able to predict not only yield and seed protein concentration, but today there's very limited work evaluating models with observed data across multi-environments on seed protein and also observed data on nitrogen fixation. So there was a pressing need to start a new team within AgMIP focused on soybean, and with the goal of evaluating and improving soybean models. And as a result, the team was created during the AgMIP8 workshop in Costa Rica in 2018. After that, we were able to get federal funds from the US Department of Agriculture to support the team activities. And in 2019, we started the first multi-model comparison. This was a pilot study with the focus of evaluating how models differ in response to CO2, temperature, water, and nitrogen. We completed this first pilot study earlier this year, and in May 2020 of this year, we started a second study focused on evapotranspiration with observed data from medicovariance experiments in Nebraska and in Canada. We are expecting to complete this study in 2021, next year. And I would like to invite anyone interested to contact me or one of the other coordinators within AgMIP soybean team to learn about opportunities to get involved as a crop modeler. And in particular, we have two studies lined up. One will be focused on nitrogen fixation and seed protein multimodal evaluation, and another looking at evaluating models under high atmospheric CO2 with observed data from phase experiments. But also there are opportunities to get involved as a data experimentalist if you have any key experimental data that can help evaluate and improve soybean models. So I would like to provide now some snapshots of our results so far. This map shows the sites that we had in our pilot study, and it had 5 sites with detailed growth data, in-season data, and in addition, 2 of the sites, Ames, Iowa, and France had data on crop nitrogen and biological nitrogen fixation. We had 3 calibration steps. We started with a blind calibration, following a very similar approach to other teams where we are providing only with phenology data in this first step. In the full calibration, we provided with in-season growth and final yield data. And in this case, we also had a third step that we call nitrogen optimization, where we provided crop nitrogen and biological nitrogen fixation. So first, we can see the results of the normal error mean square error, average from the average across models, so from the ensemble. And we can see that the crop growth variables in the figure on the left, there was a reduction from going from blind to full in the normal mean square error, but there was no difference from full to nitrogen optimization. In contrast, for crop nitrogen variables in the figure on the right, for most of the variables, we had a decrease in error when going from full calibration to nitrogen optimization. Some key findings or insights that we got so far from this study was that we had a reduction in final grain yield and in growth variables related to reproductive growth. However, we found that models had very different approaches to get there in terms of the timing of when reproductive growth starts, pod growth, seed growth, and the rate of growth. We think this is partially associated because soybean has a wide flowering and pod setting window in contrast to other crops that have a narrower window. And it also has an implication that phenology data for soybean is less useful to reduce error during calibration of crop models. Also, another insight from crop nutrition variables was that we saw that there was large variability in how models predict tissue nitrogen concentration. Especially, we saw large errors for models that had been tested less with experimental data or that were developed from cereals. So there's an opportunity to reduce error in simulation of crop nitrogen and biological nitrogen fixation when evaluating models and adjusting tissue nitrogen concentrations with additional experimental data. I want to share also this slide that shows the results from the multimodal ensemble when we build multimodal ensembles of more models, and we saw similar to found in other studies that we can reduce error with multimodal ensembles. In this case, with 4 models or more, we can reduce error during blind and full calibration. Only during full, we reduce the error below that observed in experimental trials. But we also looked at the data by location, and after imposing the restriction that the same model must be present. This is because someone might use a model and consider how adding additional models might reduce error, but the first model might not be dropped from the ensemble. And so what we found is that actually in the blind calibration, the ranking of models is very different. So a single model will not do best in all sites. Also, we find that without knowing a priori how the model will perform, a multimodal ensemble will help in many cases, but also in some cases, it will increase error. So this is a bit different from some of the results Hall was sharing a moment ago. And this was the snapshot so far of our results. There'll be more to come soon. I would like to thank all the collaborators. This has been a team effort of voluntary crop modelers that have been doing the simulations, data experimentalists that have provided us the data, and I want to thank also the coordinators, Kritika Kotari, the postdoctoral researcher working with me at University of Kentucky, and Rafael Battisti and Ken Putti. And remind there's opportunities to get involved and participate in our team. So if you're interested, contact me or one of the other collaborators. Thank you.
Thanks, Monty. That's great. Great to see the progress you made within this team, with the soybean team, which is the number 4 of the big 4 there, which is really good. I do have a question. Did you do already a sensitivity in relation to the 3 main drivers, water, temperature, and CO2? And did you find something similar as all the others found, that the largest uncertainty comes from temperature or not?
Yes. I didn't have time to share that, but yes, we saw very large differences in the response to temperature, not only to increasing temperatures, but also reducing temperatures. We see that response is not necessarily influenced by the calibration. So it's something embedded in models and how the processes are described. And it did not, that variability across models didn't change very much with the calibration.
Okay. And I'm looking at the chat, there's nobody put a question in there. If you have a question, please add to the audience, please add questions to the chat, and I look through and then we can read some out of them. If you don't have them now directly after presentation, we hope you also have some time in the end. Monty, maybe one other question. I didn't totally understand the last part where you said when you try to find out how many models you need, you found something different to what the other teams found. Do you have models which are better than your medium, or is this general finding we have also the same across the soybean models?
It could be because your results are based across sites. So if we look at this figure, one of the conclusions you mentioned were that there was not any model that will do better than the mean across models. So we saw that some models actually are also across sites. There are some models that are better than the mean or the median.
Okay.
But also, if we look at the data by location, then that effect is more pronounced, where in some cases a given model will do better than alone than when we add more models.
Okay, good, interesting. There's one question here which I'd like to read out from Dave Gustafson. Have you been in contact with commercial soybean breeders with your activity?
I've communicated with some, but not to start a collaboration yet.
Okay, great. Thanks, Monty. Thanks for the great presentation.
To share.
Yes. Our next presenter is Mark Kobielski. He is with CIRAD and CIMMYT, and he will talk about low input systems with maize. Mark, over to you.
Yes, thank you, Santold. I will share my screen. Can everybody see my screen?
Yes, we can.
Yes. Okay, so I will give you a brief overview of the progress and the remaining project we have with the low input cropping systems. I give this presentation on behalf of the whole team. I didn't put all the names, with a special mention to my colleague at CIRAD, Gatien Falconier, who did the largest part of the analysis of the model outputs. So I will start with a few words on why we launched an activity on low input cropping systems and what does it mean, low input cropping systems. And I must say that in this exercise or in this activity, we focused so far on cropping systems in sub-Saharan.
Mark, you are muted for some reason. Mark, can you unmute yourself? Oops. Mark, you are muted. I don't know how that happens. Can you try again, unmute yourself?
Yes, I unmuted, but then I get a message. The host muted me.
Okay,
can you hear?
That's not me. Okay, you're back on again.
Yeah, so a first observation and a reason why we launched the low input cropping systems activity is that fertilizer use in Africa lags behind other regions. As you can see from this figure, the average absolute rate of nitrogen use in Africa, and these are data from FAO, has been over the last 20 years about 15 kilograms per hectare, which is thus much lower than in other regions. If you look, for instance, at Europe, Europe is the green line. We have an average rate of 50 kilograms per hectare. If we look at, for instance, North America, the purplish one, there we have 75 kilograms per hectare, kilograms nitrogen per hectare. As a result of this, now I go to the next slide. Why is it not moving? Yes. So as a result of this, we have major areas on the African continent that experience nutrient limitations, and these can explain major yield gaps. The figure that you see here, or the map, is the result of an analysis done by Nathan Muller. And it shows that large regions in Africa have low yields because they are nutrient limited. And these regions, here in West Africa, East Africa, and Southern Africa, they correspond to regions where there is a quite high population density, meaning also that soil fertility built up by fallow land is no longer an option. The regions also, they are the regions where we have the cereal production systems. In the drier areas, we will have millet and sorghum, and in the more humid areas, we have maize production. So if we then look at the yields over the last, let's say, 5, 6 decades, we see that on average, yields in Africa are low, and only over the last, let's say, 10 years are increasing a bit up to 1.5 tons per hectare, whereas you probably know cereal yields in the other parts of the world are much higher and have been increasing over the last 40, 50 years. So this is why we launched this activity. What we did is we contacted several modeling groups, and at the end, we got 25 modeling groups that used their model to simulate a set of experiments in Africa. So we used the protocols of AgMIP, and with the two phases. First, a phase of model calibrations, where we collected experimental data from 5 sites with 2 experimental years, 2 seasons per site, and all the sites, all the experiments had maize as the crop grown. We did the model calibration in the 2 phases, the blind phase or the partial calibration, where we gave the modeling groups characteristics on weather, soils, phenology, and crop management, and then the full calibration, where we gave also the data on grain yield, above ground biomass, soil moisture, leaf area index, and for some sites also N content in plants. And then once the models were calibrated, we simulated scenarios of climate change, doubling the CO2, increased temperature plus 4 degrees, and we modified rainfall, 50% of the seasonal rainfall and 100% of the current rainfall. So on the sites, we looked in the literature, and in fact, we found only 5 sites where we found experiments over 2 seasons where we had sufficient data on the variables that I listed before. So that was a bit surprising, and that means also that we really have a lack of data of experiments in sub-Saharan Africa. The sites had contrasting environmental conditions in terms of rainfall and temperature, with some sites that had rainfall as high as 1,000 millimeters, while in the site at Rwanda, we were below 400 millimeters. So on the model accuracy, similar to what Montse showed, once we gave the modeling groups more data, so the grain yield, the leaf area index, soil water, then we could see that the error, the model error, was much increased, and especially for grain yield and soil water content. So I think this is an observation that we can see all over the modeling exercises, maize, rice, wheat. So there is no surprise here. I want to show you this slide, and this is maybe one of the main results, and this graph gives you an idea about the impact of a simulated increase of 4 degrees. And what you see is the results of the model ensemble mean. So all the 25 models of an increase of 4 degrees compared to the baseline. And the model outputs were also averaged across the 5 sites. So you can see that we have a relative grain yield change of between -10 to -25%, depending on the nitrogen inputs, on the nitrogen fertilization. The higher the nitrogen fertilizer, the more decline in relative grain yield. For the details of this exercise, I refer you to a paper that was recently published in Global Change Biology. So just to give you an overview of the main results of our study. So we found that the grain yield was relatively accurately predicted by the different models, and that we had lower accuracy for other model variables. We also found that the maize and intensively managed with more N fertilizer will be more sensitive to climate change. I think this is an important result. That means that the design of the adaptation options or adaptation practices to climate change for farmers in sub-Saharan Africa will modify or will depend on the degree by which they intensify their crop production through a balanced nutrient management or a balanced fertilizer applications. And then more on the technical side, we also think, or we found that crop models that are explicitly accounting for N stresses and N leaching, that are simulating on a daily basis the soil N processes, they show stronger interactions between climate change and the input of N fertilizer. So another feature or another outcome that we found from this exercise was that there was a huge heterogeneity in the simulated model responses by the different models. So this shows the simulation of the grain yield as a function of N fertilization and input for the 5 sites by the different models. So you can see that there is a huge heterogeneity. We are still looking and trying to understand why there is such a high heterogeneity, and maybe we can use that also to say something about possible model improvements. And now a few slides on the new phase that we are launching on the long-term simulations or in the case of low input systems. We think that the low input cropping systems, we think that the greatest importance for good simulations is looking at soil fertility and looking at the supply of nutrients, in particular N, but maybe also phosphorus. So to us, the challenge is to simulate the annual N supplied right over multiple years, and this without a reinitialization of the model each year. And therefore, we will look at long-term interactions between the crop and the soil in terms of soil carbon and nitrogen cycling. And therefore, in the new phase, we set the objectives, or we have the following questions. So what are the needed crop model improvements from the different model intercomparisons? So again, we will go to the 2 phases of the model calibrations under current climate, as we did in the previous exercise. And herefore, we have identified experiments, 2 experiments in Kenya, or 1 experiment in Kenya at 2 sites, on a clayey soil and a sandy soil, and the same in Zimbabwe, 1 experiment on 2 sites, a clayey and a sandy soil, where we have data on crop yield, soil organic carbon, in some cases soil moisture, and in some cases also plant and uptake. What is missing probably is the soil and supply, but we are trying to get those data or to measure those data over the coming year. So then the second question is, once these models are calibrated against those data sets, what are climate-driven changes in crop yield over the long term and in soil organic carbon contents. So in this way, we can look at climate change effects on yield and on soil organic carbon, meaning that we can also look at mitigation. And the question then is, in how far do our models agree? What are the differences and why are there differences? So we will apply also defined scenarios of CO2 changes, temperature changes, precipitation changes, and combine this with different levels of N fertilization. And that was it. Thank you for your attention.
Thank you very much, Mark. I think it's very important component within AgMIP, looking at these low input systems, because there are many around, and often it has been suggested to intensify them for increasing production. But what you showed, I thought was very important, is that with intensification, with higher yields, you make these crops also more vulnerable to higher temperatures. So it's there must be probably a balance somewhere to how far you intensify and how vulnerable you become with that. So there are a couple of questions in here. One was, there's a model mentioned, it's called QUEFTS, which is obviously a very widely used one in Africa. Why hasn't that been used? Are you aware of this model? And yes. how did you select models for your team?
So the selection of models, we went back, since we had maize as the crop, we went back to the maize group and asked to give the contacts of the people that were modeling the exercise of maize, where they were using data in Brazil, 4 sites in the world, Brazil. I don't remember exactly the sites. So we had a list of those modeling groups, and then we looked in the literature also, the modeling groups that simulated cropping systems in Africa. So that's how we got there. Now, why we are not using QUEFTS? Because QUEFTS is not a mechanistic crop simulation model. QUEFTS is quite different. QUEFTS uses empirical relationships to derive and supply phosphorus supply and potassium supply from soil characteristics, and uses then the relationship between uptake of those nutrients and grain yields. So this is quite a different model than the ones that we have been using.
Okay, great. There are actually a lot of questions in here, which Mark, I think we need to move on to the next speaker. I think we come back to in the general discussion to it. I hope there's actually a lot of more questions in there which we cannot go into at the moment. I think some of them related to uncertainty and in relation to nitrogen modeling, you might have addressed already. Let's park them for a moment. And I'd like to introduce, thank you very much, Mark. I'd like to introduce the next speaker, Dave Gustafson. He's with Agriculture and Food Systems Institute, and he will talk about modeling fruits and vegetables. Dave, over to you.
Thank you, Sentold. I assume and hope I'm unmuted. Is that correct?
Yes, we can hear you, and we can see your slide.
Okay. Do you see me?
We can also see you.
Okay, great. Well, thanks very much for the opportunity to present here, and thanks to you also, Sentold, is serving as co-lead for this project, which has been ongoing for quite some time. And as you know, and I think others may have heard, we've been looking at climate adaptation and mitigation opportunities in United States fruit and vegetable supply chains. So just briefly today, I'm going to do a little bit of introduction, some sample results, conclusions, and what's happening now and next steps with this project. So we're obviously shifting gears now away from the staple crops to talk a little bit about fruits and vegetables. As I think you all know, these are very important to include in our diet. And we are seeing increased demand for these foods, and especially foods grown locally for all sorts of reasons, particularly here in the US. but we also know that these crops are subjected to the potential for increased threat due to climate change and competition for natural resources, as well as competition for labor. Fruits and vegetables here in the US require the use of a lot of labor. Some of that labor is coming from other countries, and obviously there's been constraints on that lately. And there's a huge drive within the industry towards automation, which is one of the big things that's happening. And the other thing about fruits and vegetables here in the US is that most of them come from California today, something like 80 to 90%. California, as I'm sure you've all aware of around the world, parts of it are on fire. Thankfully, not really impacting much the fruit and vegetable supply chain at this point, with the possible exception of grapes that go into wine production. But there is nevertheless significant overcommitment of groundwater and other sources of irrigation water, particularly in the southern part of the Central Valley of California. And so over time, there's going to be increased pressure on growers in those areas to justify the use of water, and perhaps even have to pay for it in a way that historically has not been necessary. So it is among those big drivers that caused the interest in this project, which, by the way, does receive funding from USDA NIFA, which we're very appreciative of. We have a multidisciplinary team working on this project. There's not a lot of us, but we do have a diversity of expertise, and we come from a number of different institutions. I've already mentioned Central from the University of Florida, but we also have a number of collaborators from here in the state of Washington, which is where I now reside, Washington State University, as well as the University of Illinois, University of Arkansas, IFPRI, and then WAYS. Some of you might not recognize the WAYS acronym down here with the economic modeling team, but that's primarily John Cruz and his team, who performed a lot of the economic modeling that are associated with results that I'm going to share here today. And then one comment about my home institution on this project, which is the Agriculture and Food Systems Institute. That might be a new name to you, but it is in fact the new name of what used to be called the ILSI Research Foundation. Same legal entity, but a new name. So just a little bit about our methodology here. We had a crop modeling ensemble, and we did that modeling for a number of counties throughout the United States that are important for fruit and vegetable production. Into that modeling, we had 5 GCMs, and we used a high greenhouse gas emission scenario, RCP 8.5. We looked at current and future scenarios with and without adaptation, primarily shifting and planting date for the 2030s and the 2050s. Those crop modeling results were then fed into some economic models, and there was some back and forth. And I don't have time today to go into all the details, but we did develop an approach to look at both the climate change component to yield gain, as well as the technology component to yield gain, based on observations concerning USDA average yields for these crops. And I am so sorry, my computer has just done something strange to me. Are you still able to see my screen?
Yes, we are.
Okay, sorry for that interruption. What we then do using the economic models is we calculate land use change, as well as changes in the domestic amount of production and prices for these crops. And then in addition to that, mitigation scenarios. And the mitigation scenarios, that is climate mitigation scenarios, are coming out of life cycle assessment modeling, where we looked at both carbon and water footprints for these crops. And I should comment that all of the results at this scale are at the scale of the crop reporting district, which are basically groups of counties here in the US that have been defined by USDA. So there was some geographic precision to our results. So this is some sample results for potatoes. Here in the US, potatoes come primarily from up here in the Pacific Northwest, but they are produced across the country as indicated here. And we generally, and this was somewhat surprising, but we found rather modest climate effects on yield, and they were actually mostly positive, which I know runs contrary to some of the other results we've seen for other crops today. But we found that the heat stress that could be happening is avoidable by shifting planting dates, which has already been happening here in the US, and relatively minor changes in total production area with some shifts towards those regions which have higher yields, such as right here, where I happen to be speaking to you, is this region right here in the state of Washington. We also looked at revenues using the economic models done by John Cruz's team, and found that in general, revenues for growers were steady to slowly decreasing, but there was a lot of regional variation in profitability. And again, in these very high yielding areas up here in the Northwest, we saw higher per acre yields. One of our other interesting results, and I guess it's no surprise to many of you on this call, is that water use efficiency will increase over time in terms of yield per amount of irrigation water used, primarily to the effect of higher CO2 levels, which essentially makes the plants more efficient on their use of water. We also did detailed life cycle assessment work, looking at not only the production part, in other words, the on-farm part of the activities to produce foods. The two processed foods that we looked at in this first application of the methodology were a form of processed potatoes, which is actually frozen French fries, but we also look at processed tomatoes in the case of pasta sauce. And what we found is that in terms of carbon emissions, there's quite a lot that's coming from the processor, the retailer, and the consumer that contribute to the overall carbon emissions, GHG emissions for these foods. And so there's actually significant mitigation opportunities later in the supply chain, which we do explore in the paper. And both the land and the water footprints will decline over time as yields and water use efficiency continue to increase. So just to kind of wrap it up here, we're trying to be brief, obviously. The team has developed a novel approach to looking at climate adaptation mitigation options in fruit and veg supply chains. We found that there's resilience to these systems unexpectedly, and that there are some opportunities for mitigation, and that there will be significant improvements in both technology and expected yield increases. So next steps, a paper is describing these results on potatoes and tomatoes has been submitted for publication. We're now using this approach to look at a series of other fruits and vegetables here in the US, but we believe that this method is applicable to many other such crops and in other geographies, and we think that it could be used to help better inform decision making by all sorts of players in these supply chains, which hopefully would help improve our diets globally if we were to ensure sustainable and greater consumption of fruits and vegetables. And with that, I will just say thank you once more for your attention and turn it back over to Senthil.
Thanks, Dave. That's very interesting. I think it's also very sort of appropriate within AgMIP to see how this session is about crop modeling, but it's about an approach to bring other modeling approaches into a team to look not just at impact and adaptation, to be also now able to look at mitigation. Are there any questions for Dave? I can't see any, so please sort of add questions to the chat, and we probably come back to this presentation during the discussion. So thanks, Dave, again. Thanks for this very interesting presentation. Our next presenter is Lev Siska. He's with Columbia University, and he will talk about nutrition, diets, and food security. Lev, over to you.
Apologies, I'm looking for my presentation. Okay, so I'm going to try and give a sort of a history. Can you see this okay? Can everyone hear me?
Yes, yes, yes.
My sincere thanks for the invitation to talk. I've been working on the issue of carbon dioxide and nutrition for a number of years, and I'm going to give you sort of a very rapid overview of this. It's going to be difficult. Think of it as sort of trying to
if we cannot
explain Game of Thrones using Twitter. I mean, it's done at the International Rice Research Institute back in the mid-1990s, where we were looking at carbon dioxide levels of +200, +300, and also looking at temperature. And one of the things that we found, to our surprise, was a consistent decline with rising carbon dioxide in terms of rice protein. And this was very much in line with what Jan Conroy was doing back in Australia at the time, showing that for both nitrogen and phosphorus, it was affecting nutritional content for a number of cereals. To sort of update you, this is some more recent work done for an evaluation of about 18 different lines, both japonica and indica from Japan and also from China for the various rice hybridizations. This is looking at it from the point of view of 18 different lines, again, a fairly broad genetic basis for this, but looking at a 550 part per million relative to ambient, where we see a fairly consistent decline occur in this century. Now, let me kind of change it a little bit, because we always think, well, it's the future, it's the future. But in fact, CO2 is about 30% more now than it was back in 1970. So this is some work that we did looking at spring wheat, looking at lines that were released throughout the 20th century. And you have lines in 1903, 1921, 65, 96. And you can see that under sort of current CO2 levels, that there is, with hybridization, a decline in protein. But also note that the CO2 aspect and part of that is also declining in terms of the protein levels as well. And this is some work for rice. Shoot, I'm going to go back here. Sorry about that. There we go. This is some more recent work for rice, looking at the change in protein concentration for
Sorry, can I let me interrupt you?
Again from 3.
Left, can I interrupt you? Sorry about this. Your voice is sort of coming in and out. Maybe you should switch off your video to maybe getting more bandwidth so we just hopefully get your full voice. There's a lot of cut out of your voice there.
I apologize. My internet connection here is a little bit tenuous. I'll try and do that. Let's see if we can go forward. In any case, this is that, and we want to go in terms of overall. This is some work from Daniel Taub, looking at barley, rice, wheat, soybean, and potato. This is the percent change under a doubling of elevated CO2. And with the exception of soybean, which of course is a leguminous plant, we see a consistent decline in terms of protein concentration. This is some meta-analysis by Irakli Lalaz, looking at all of the different changes with respect to the plant stoichiometry. And again, you sort of see the decline and also the fact that carbon is going up relative to the other issues. I want to add a little bit more. I'm working with Weilu Wang, who is a collaborator on the T face, the temperature-free air CO2 enrichment face, which is shown here, to try and address the question of whether or not temperature has an influence, a synergistic or antagonistic influence with regard to CO2 effects. And this is a breakdown of the different amino acids, looking at ambient CO2 and ambient temperature, ambient CO2 elevated temperature. But the elevated CO2 elevated temperature relative to the elevated CO2 per se. And the point of this is just simply to say that rising temperature, at least in this instance, does not compensate for the effects of CO2 with respect to amino acid composition, both for the essential amino acids and the non-essential ones. So we don't see much change here, at least with rice, in the context of both CO2 and temperature increasing simultaneously. I want to kind of emphasize that it is not some work by Augustine and Blumenthal showing a 7-year change with respect to percent nitrogen, which is a surrogate for protein. Again, seeing that regardless of temperature, increasing CO2 can reduce the protein concentration and as a result, decline in protein as well. And this is some data that we have for pollen, for goldenrod pollen. The pollen was obtained from the Smithsonian historically, looking at pollen that was collected from goldenrod back 100 years ago, and looking at the estimated protein based on the nitrogen composition and the subsequent changes and the carbon-nitrogen ratio. And we can see here that it's not just for food that we eat, but it's also food for bees. And this is some experimental work looking at a CO2 gradient tunnel for goldenrod as well, showing a similar, slightly different numbers, but a similar change. Now, this in terms of what happens to pollinators and how these can be modeled and how can these be forecast, to my knowledge, nothing of that is in fact happening. Is it more than protein? Yes, we see a number of elemental changes, although there's a little bit more variability in there. This is again for rice for the 18 different lines, looking for iron and also for zinc. But you can see that there are some lines that are not showing a change in iron and zinc. We don't know the basis for why that is. And obviously the more work needs to be done in that area. When you look at rice CO2 and nutrition, we can go a step further and look at some of the changes in the vitamin composition. Here for the Chinese varieties, looking at vitamin B1 through vitamin B9. And again, seeing a consistent decline in the concentration of those vitamins for rice. So it's to my best of my knowledge, the sort of integration of protein, elemental changes, and vitamins has only been done for rice in the context of CO2. We looked at one vitamin which was really interesting, vitamin E, alpha tocopherol, and what we found is that for the most part, there was no significant decline. In fact, overall, there was a significant increase for rice for this particular vitamin. And we were wondering why this is. Why are some vitamins going down, others going up? And we came up with a hypothesis. We'll call this the firm possibility of a definite maybe. And what we're looking at here is the percent change, the increase or decrease relative to ambient carbon dioxide as a function of the molecular weight. That is to say, if you're seeing this change in carbon to nitrogen, then those elements that have no nitrogen, that are high in carbon, such as tocopherol, actually seem to be going up, whereas the more nitrogen a particular secondary compound has, the more adversely it is affected by the increase in CO2. And again, this is a working hypothesis. We've seen, since this time, we've seen some other data looking at caffeine, for example, in tea and in coffee, and seeing that caffeine concentration, caffeine is a bicyclic alkaloid with a lot of nitrogen in it, it is also going down, consistent with what we see here. So modeling all this, as you can imagine, is something of a nightmare. This is looking at it from protein, iron, and zinc, using some of the work that Rocco Velaz has. Sam Myers has a similar model. And I won't go through all of this because it's really complicated, but this is just looking at it from the point of view of inputs in terms of protein and the various elements. The Everest, if you will, of modeling in this case is how do you then model these inputs and then relate them back to health consequences? And we're far away from where that needs to be. I will just point out that this is recently came out in Lancet Planetary Health, if you'd like to find out a little bit more about it. So in summing up nutrition, there's ubiquitous data indicating shifts in carbon and nitrogen, and carbon to other elements, that there is this stoichiometric imbalance. The atmosphere is increasing in carbon, but the soil is not keeping pace with it. The mechanistic basis for why this may be, too much of one, not enough of another, other different physiological parameters that need to be investigated. is still underway, but so far looked at. And I also want to emphasize that this goes beyond just a human perspective, that this is part and parcel of what's happening to the food chain overall. And I think one of the challenges we have is to look at it in terms of other aspects of agricultural production. So if in fact honeybees are going to be impacted by a change or CO2-induced change in protein, what does that mean in terms of their efficacy, given their importance? The research has implications beyond that. Modeling is going to be really tough. Whatever epithet you want to use here, whatever four-letter word you think is appropriate, it's going to be difficult to try and model all this in the context of just the human food chain, but also in terms of other food chains as well. Are there other impacts in terms of food allergies? We just simply don't know. So all of this is to say that there's some really interesting data, lots of interesting stuff going on, but in terms of what that means with respect to trying to model it and what the impacts of it are going to be, we have a long way, everyone. If you have any questions and we don't have enough time for them, please feel free to contact me directly. So thank you all very much.
Thank you very much for this very interesting presentation. And it's just so important. Yes, there are a lot of modelers in here, and we spending a lot of time looking at yield and ET, and you saw we just stretching, getting into grain protein, but there's so much more which we need to understand to sort of really talk about food and food implications and how a crop in the field might be influenced in this way. So I think you touch really important aspects. It's the other minerals in there, it's vitamins, and as you said, I think when it comes to allergies, it might be even getting more complicated of how we do it. Do you have any suggestion maybe of how modelers should address that? I'm sure you know a bit about modeling. Should it be done more through a mechanistic approach, or would that be some sort of an add-on, might be a fast way to get some progress in there? Any ideas there, Lev?
I think when it comes to modeling, I think there needs to be a removal of the silos where we can go to the health community, the public health community, and to begin to interact with them in terms of trying to understand the role that this plays in terms of human nutrition. As Naomi Fukugawa mentioned, the greatest threats from health-wise are now related to nutrition. And so trying to make that connection, trying to make that link will provide some impetus, I think, for the public to understand why that is important. But I also want to sort of reiterate what was being presented for some of the other crops, the other vegetables. We know a lot about the big four, as you mentioned, but there's a lot in there that we don't know, and we really need to find out more about those in order to integrate what the overall effect is going to be.
Great. There's a question from Suang Kim. He asked of how could you separate the effect, trace things back to just CO2 in relation to protein. I guess it applies to others as well. Why could you say this is poorly related to an increase in protein in some of your data?
I'm sorry, Senil, you're fading out. Can you repeat that?
So there's a question of how could you contribute the change in protein in the past to CO2 only, since there are a lot of other things have been changing as well.
Yes, and one of the major issues has been that hybridization, as breeders have selected for faster growing species, that increased growth per se will decrease protein levels. So we have two experiments that sort of say that CO2 by itself is causing this. The first is, as I showed you with spring wheat, we looked at all the different lines, or not all, but we looked at different lines that were introduced during the 20th century. And you can see that what hybridization has done is lowered the protein content, because when you want wheat to make certain kinds of pasta or certain kinds of bread, protein isn't always something that you want. But regardless of the variety, all of them show a decline in pricing CO2, both recently and projected. The second one is, to my knowledge, no one has done any hybridization on goldenrod pollen. That is separate from human endeavors. the fact that we can look at both historically and experimentally that CO2 is causing that protein decline suggests that it is more than just management, it's more than just breeding.
Thanks, F. Very, very interesting, very important. Again, I stressed it before, the collaboration of modeling and physiologists and experimentalists is just so important to really make progress in this space. So thanks again for this great presentation.
Thank you.
The last presenter in our, last not least presenter in our panel is Cheryl Porter, my colleague at UF, University of Florida, and she will talk about advances in data interoperability for crop modeling. Cheryl,
okay.
Over to you.
Now for something completely different. So this topic has to do not so much directly with modeling, but how do we make data available for modeling? And specifically, how do we make some of our legacy data sets interoperable such that they can be interpreted in an automated way? So there's a lot of partners in this project. Most recently, we've gotten funding from USDA NIFA. I've got all the funders on the ending slide, but we've also been working with the CGIAR Big Data Platform and National Agricultural Library in the US, and also IFDC, the International Fertilizer Development Center. So the problem is that after a research project is complete, the data are often lost after the research objectives have been obtained. And so we're looking at ways to make these data available, particularly for quantitative type analyses. So the idea is that if you could gather these data sets from various multiple environments, different kinds of management, different genetics, and put them all together such that they could be combined into a metadata set, they could be useful. So as a group of modelers, I can challenge you, and I'm not going to even answer the question, but what would you do if you could obtain data from research projects that had already been completed, and you could combine the data across management, environments, and genetics? There's so many answers to this question. So what we're doing with ARDN, and this is Agricultural Research Data Network, is we're starting with AgMIP data tools that were developed under the DFID funded project for ensemble modeling for regional integrated assessment. And we're building on those tools to allow us to bridge the gap between the supply of data on one side and the demand for data on the other. Knowing that the challenges are that these data sets are in different geographic locations or in different databases, they use different formats, vocabularies, and so on. So how do we do that? So stepping back a little bit, the data interoperability tools that we started in the DFID project had to do with taking data from multiple different data sources, bringing them into this harmonized AgMIP format, and then making them available for multiple models. And then finally bringing model outputs together into a harmonized format. And along the way, we built in this idea that not even very detailed field experiments may not have everything that a crop model needs. And so you can supply supplemental information and keep that supplemental information separate from the raw data. So this harmonized data format has been adopted by IFDC, International Fertilizer Development Center, for use in their data standardization, access, and archiving. We've worked with CGIAR on their platform for big data to use some of these AgMIP methods to harmonize their data within the Guardian data portal And we're currently also working with USDA, National Ag Library, as part of this NIFA FACT grant to do something similar with their Ag Data Commons portal. So the concept is that, okay, I'm using on this slide an example of the Guardian platform. So this is a data and document sharing portal for the CGIAR system. So they've got on Guardian already their core metadata. And we're going to supplement that with what we're calling these sidecar files. So this is annotation of the data that allows us to interpret the data in an automated way. And these data sets are actually sitting out in remote distributed data locations. So in this example, I show Harvard Dataverse, a CIMMYT database, something that's been published in the Open Data Journal for Agricultural Research, and Ag Data Commons. And as long as we can describe those data sets in a way that allows us to understand the contents of the data, we don't have to change the raw data. It's sitting out there in a raw format with a DOI, and it's sort of sacred. We can't change that. But we can provide the means to translate it. Yeah, so that's, so the three different sidecar files, one is to map dataset variables to known ontologies so they can be discovered through the semantic web. The other is this roadmap for translation to AgMIP format. And the third is actually a subset of the data that allows rapid searches to be done on the data themselves, such as, you know, ranges of dates, or whether fertilizer has been added, or whether it's irrigated, or maximum yields, you know, so on. So you can search, do these advanced searches on the data without actually accessing the raw data that's sitting back here in these remote data sets. So here's an example of a data set that came out of Guardian. This is Tomasa taking maize agronomy to scale in Africa. It's in a spreadsheet, so it was fairly easy. This is actually low-hanging fruit. It's a very well-organized data set. Here's an example of two little snippets from a sidecar file too. So this is the roadmap for translating the information that's in that spreadsheet to AgMIP format. And it tells you how to, you know, the URL for accessing the data, which worksheet in the data we're working with, and individual columns and how they map to AgMIP standard variables, which use the ACASA vocabulary. And one of the really hard parts of doing this is to create that sidecar file too, to annotate the data such that we can understand how to automate translation of it. And we have a tool for that. And this is something that is new within the last year, something that we developed as part of the NIFA project. And it allows you to visualize this spreadsheet and then map each column of data to a particular ACASA variable name. So it's still a lot of work, but it's a lot less work than it used to be. And here's some of the flows of data. So you can imagine that, you know, with this VMapper tool that I just showed, you know, that there's a workflow for people who want to supply their data to the Arden network. Okay, so now it requires some annotation of their data set such that it can qualify for inclusion in the Arden network. We have end user data tools. So this is where our traditional AgMIP translators reside, where we go from the standard ACE-B. ACE-B stands for AgMIP crop experiment format, and B is binary. And then these are the translators that we used in some of the AgMIP multi-model exercises, which allow us to go from ACE-B to the various specific model formats. And then we have, that we're working on, is some search and discovery tools that allow us to take subsets of the data and use them for advanced searching. And again, that is the AgMIP part of this. So I think I've covered most of this that we're working with National Ag Library, CGIAR, and IFDC. We have just started putting together a website that describes Arden and the tools and methods. So there's quite a bit of content there, and hopefully in another month or so, there'll be quite a bit more. So if you're interested, you can check it out there. And here's a lot of the partners that are involved in these various pieces of this Arden project. And thank you for your time and attention.
Thank you, Cheryl. That's again a very important aspect of AgMIP, where we want to work together. We need to share data. The collaboration is often about sharing data and getting access to data, and having you and your team sort of looking at ways to help making this exchange or the access to this data more easy. That's, I think, a very important task. If someone wants to get access to these tools, what do you recommend, like a certain group, maybe one of these groups talking today, what do you suggest there? Yeah, so all of the AgMIP tools and all of the Arden tools, for that matter, are open source. They're freely available. We have AgMIP tools available on the tools.agmip.org website. Most of those have been there quite a while. The new Arden tools are still under development. If somebody is interested in trying them out, we would be more than happy to let you be a guinea pig. But and actually, I should mention, so the Arden project is a 3-year NEFA project. We are just at the beginning of year 2. In year 3, so a year from now, we will be opening this up and trying to bring in a larger group, and we will draw from AgMIP for sure to fill that out and start, putting the word out there and letting people try to use these tools, and hopefully filling out the Guardian and Ag Data Commons portals with Arden compatible data sets. Thanks. So to everyone, if you're interested in that, I think most of the modelers are, talk to Cheryl, get in contact with her. There's a question here from Jurian Pullens. Are there some crucial ontologies that you work with, Cheryl? Yes, so yeah, I kind of glossed over that. So the AgMIP vocabulary, we have adopted the ACASA data dictionary. ACASA being International Consortium for Agricultural Systems Applications, which doesn't even exist anymore, but the data dictionary lives on. And so that was originally developed to be a documentation of field crop experiments. We have expanded it for use in AgMIP, and we're continuing to expand it, especially as we work with IFDC, where they're collecting all sorts of micronutrient type data. But we've also mapped ACASA variables to crop ontology, agronomy ontology, units ontology that are being used especially in the CGIAR system. Great. Okay, I think again, thank you very much, Cheryl, for this very interesting, important presentation. With that, I think we close the individual presentation, but I'd like to open now up for more general discussion. I've seen there have been a lot of questions actually on there. I can't actually go back to all of them, but I also noticed that some of them have been answered. If you still have some more general questions, more for all or for some of the presenters, please maybe ask them again. We have about a bit less than 20 minutes, 15 minutes for some discussion to maybe dive a bit deeper in there and try to tease out some of the links. Again, we had Monty Salmon talking about soybean modeling, Mike Kobelis talking about low input systems for maize in Africa, Dave Gustafson about modeling food and vegetables by also expanding into mitigation, Lev Siska about data, about understanding nutrition diets, and right into vitamins and other aspects which are actually very important for diets. And we just heard from Cheryl advances in dealing with data and helping us building links by also getting better access to data. So are there, from the audience, are there any questions which are maybe more general or still some specific to some of the speakers? So there's a question from Dave Gustafson. Are there links between crop modeling and any geochemical modeling efforts with AgMIP and the important soil carbon issue? Is there someone who sort of wants to jump in there? All I can say, there's actually two activities based on around soil carbon and rotations. One led by Bruno Basso, where there has been actually some modeling, and there's also a paper coming out how important it actually is to track the carbon in the soil and the interactions back in terms of how much water they have and back to the yield as well. Anyone else wants to jump into soil carbon issues in relation to AgMIP?
Yes, this is what we now plan to do, is looking in how far the different models, crop soil models, can simulate soil carbon sequestration over the long term. And herefore we have, as I said, two sets of experiments, one in Kenya and one in Zimbabwe, where we have observed data on soil carbon as a function of different treatments. And the treatments are addition of different sources or different qualities of organic matter. In Kenya and in Zimbabwe, it's more around treatments on conservation agriculture practices, including mulching.
Great. I also mentioned there are actually two activities already on this in AgMIP, and I just see Klaas Nendel in here, and he might want to comment on his particular activity where his group actually is looking at soil carbon as well. Klaas.
Yeah, there's the global microresonator network going on, and there are up to 15 sites in the world where we're actually measuring nitrogen mineralization. This will feed into AgMIP in a few years, and this mineralization, the soil organic matter turnover, will then also directly link to the carbon turnover. So we'll observe nitrogen and learn about carbon with that, and this will help us to improve model. There will be a very nice data set out there.
In relation to this, there's a question from Mukhtar Ahmed in relation to grassland modeling. What is happening in grassland modeling? I've seen actually activities which I think were originated, were started by Jean-Francois Susanna. There's actually a group, but I think it's mainly in Europe, working with quite detailed data on above and below ground grassland modeling. I'm not sure if anyone in the audience can give us some more insight onto this. Klaas, maybe you again.
Yes, this is exactly what they did. However, I learned yesterday that they were also looking into croplands, gaseous emissions from croplands. But in fact, they started with grassland, and there are some papers on grassland activities out there. In addition to that, the MAXUR, sort of the European hub for crop modeling, they also had a grassland pillar. And also there were a few papers published also on grassland modeling, which eventually could have another link to AgMIP later on.
Great, thanks, Lars. Here's a question for Monty. So we've seen the impacts of CO2 and temperature on minerals, protein, vitamins, and so on from left. And we also seen that for protein, soybean didn't respond. How do you deal with that in your modeling? Is that part of it yet? Are you there yet? Do you look at that? Do you have any feeling of how the models deal? Maybe just start with protein and soybean.
Is this question written in the chat? Were you reading from the chat?
No, I made it up for you.
Oh, okay. Sorry, I was trying to find it and I got distracted for a moment. Can you repeat? You were saying.
Lev showed in his data there were soybean not responding in terms of protein to higher CO2. I'd like to hear from you, since you also looked at CO2, maybe not with data yet, but with in the sensitivity analysis. What do the soybeans models do with higher CO2 and maybe also with higher temperature?
Yes, so what we see is that in most models, the tissue nitrogen concentration, both in vegetative and the seed protein, is quite stable. So has little response to changes in CO2, except for some models that some show that increase and some show, a couple of models show a steep decline, similar to what a cereal will do, and it's because of being developed from cereal, but most models show very little variability. In terms of the temperature effect, actually, I believe one of the models only considers temperature effect on protein concentration. Other models do not consider a direct effect on temperature other than the indirect effect on carbon growth and nitrogen demand.
Good, thanks for this. I have another question which I picked up there. I think it was answered, but it might be important to come back. Dave, it's a question for you regarding the multimodal ensemble. More clarification, but it might be important for this audience to sort of clarify at which stages the approach you presented, Dave, had actually multimodal ensembles in there.
Sure, I answered it online to Cheryl, but basically I had a slide on this, but went over it way too fast. Sorry for that. But yes, we did have multiple crop models. We also had multiple GCMs, and therefore, in a sense, multiple climate models feeding into the process. So we ended up with quite a sizable ensemble, especially in the case of potatoes. I believe Central, something like 5 crop models, 6, depending on how you count them, I guess. Yeah. And then fewer on tomatoes, and most likely fewer, but still multi-model is the approach for all the other crops and for all the reasons that we're talked about elsewhere for the staple crops, which is that the median of any ensemble of models is basically, statistically speaking, always going to give you a more reliable approach. And so, yes, we use multiple models on the 2 vegetables we modeled so far, and we will be applying it throughout the rest of this project. And I think it's certainly good modeling practice, and as this team obviously very well knows, to be using multiple models whenever possible. And if not possible, then you probably ought to be thinking about getting a new model into the mix, as we had to do actually with some of our crops that we looked at.
Thanks, Dave, for clarifying this. There's a new question on here from Dan Williams. She asks, is there anyone looking at modeling regenerative agriculture compared to conventional agricultural models? I'm not sure actually if there are different models, but it might be something Mark could comment on, because the function is the same, but the approaches of managing agriculture might be different. Mark, do you want to comment on this?
My question is, what do we understand under regenerative agriculture?
Dan Williams, can you clarify that? My feeling it's a more conservative approach to agriculture. Again, probably low input and so on.
It shows up everywhere now, but I'm not sure whether it's well defined. What we can do is use the models to simulate the effects of conservation agriculture, meaning the effects of no tillage with mulching, and to a lesser part the effects of crop rotation, because it's not always easy. Rotating crops have different effects. One of the effects is the nutrient cycling. I think most of the models can handle that. in a certain way, but the effects of breaking pest cycles, that I don't think that there are a lot of models that can take that to, or do take that into account.
Okay, so there's a bit more clarification. It's about conservative efforts. I think again, most of the models we have which try to do the dynamics, and if they don't go into areas which the models don't deal with, for instance, pests and diseases, then you can actually use these models also to explore the effect of conservation efforts. And I think there's actually something going on. If there's a particular group within AgMIP, I don't know, but I know colleagues working in this space, and also now it's possible to use many of the models we have in this space. I do have another question for Cheryl, because we talked about quality aspects of crops, but there's another quality related to data. Does your system also help with checking the quality of the data which are available?
We do not make any attempt to rate the data. However, if you can put the data into a common format, you can do things like look at the numbers of, measurements of leaf area index, or, grain yield, or those kinds of things. That's not really telling you about the quality of the data, like how carefully it was measured and that sort of thing, but it does tell you how, how detailed that particular experiment was and what the variables that were measured were. We purposely did not want to put rules on the data. We're just saying if there are data that can be mapped to these terms, great. Let's provide a means of doing that. It's always going to be user beware, and the way that we're doing things now, preserving the raw data while we make interoperable data available, allows somebody to go back to the raw data and dig out some of the rich details, like that we had a pest infestation, in June of 2015, and therefore our yields were low. And we don't necessarily have quantitative variables that can capture all that, but the raw data is all there in its complete form.
One thing regarding to quality I wanted to suggest here, which many of the AgMIP teams do already, is actually working with the people collected this data to really understand what's behind, understanding how they were measured. There are different ways to measure the same thing, to ensure that the modeler fully understand what these data are. That's another way, I think a very important way to build the team, to be inclusive, take the experimentalists into the team, but in this way also ensuring of how these data are used to get most out of them. With that, I think I'd like to conclude. I think we have a few minutes to actually get out of this link, and you have to link again into use the other Zoom link to get back to the main group. I'd like to thank all the presenters. I think they're really great presentations, gave us, I hope also to the audience, a great example of what's going on. There's much more going on in AgMIP, but I think we learned something what's happening at different parts. I hope it encouraged you to sort of contact some of your colleagues and get involved. With that, a big thank to the presenters, also a big thank you to the audience for some very constructive questions. And I hope to see you all back again in 3 minutes in the other room there. So you need to link out here and link back into the other room. Thank you.
Thank you.